Hello and welcome everybody to my channel MSc Entrance, and today we will be discussing GAT B question paper conducted in the year 2022. If you are new to my channel, please do like, share and subscribe. Then next one, match items in list 1 with items in list 2. Inactivated vaccines. Inactivated vaccines based on the killed or altered disease causing germ. So A2 is found in 3 cases, 1, 2 and 4. So option 3 can be eliminated. Live attenuated vaccines based on the weakened form of the pathogen. B3 is found in 2 and 4. So option 1 can be eliminated. Then conjugate vaccines. Conjugate vaccines are based on the combination of the weak antigen coat of the pathogen and strong carrier proteins. So C4 is found in option 4. So option 4 is your answer. Toxoid vaccines are based on the poisonous protein made by the pathogen. So the answer here is option 4. The next one. Which of the following are professional antigen presenting cells? Professional antigen presenting cells are B cells, macrophages and dendritic cell. They express MHC class II molecules. So the answer here is dendritic cell. Answer is option 3. Then given below are two statements. One is labeled as assertion A and the other is labeled as reason R. So assertion A, polyclonal antibodies bind to the same antigen but recognize different epitopes. That is correct. Polyclonal antibodies are produced by different B cell clones in the body. Yes, that is why they can recognize different epitopes. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then identify the statement that is not applicable to enzyme catalyzed reaction. So the reaction proceeds with the conversion of the substrate to a high energy transition state. Enzymes alter equilibrium constant of the reaction. That is not correct. Enzymes do not alter the equilibrium constant of the reaction. Then next one. Match items in list 1 with items in list 2. Cytochrome oxidase. Cytochrome oxidase is for copper ion. So A4 it is found in 1 and 2 only. So 3 and 4 can be eliminated. DNA polymerase has got magnesium ions, nitrate reductase has got molybdenum ions, and urease has got nickel ions. So the answer here is option 2, that is A4, B3, C1 and D2. Then next one. Which of the following amino acids are both glucogenic and ketogenic? So out of these, isoleucine, tryptophan and phenylalanine are both glucogenic and ketogenic. A, C, E only. So the answer here is option 2. The next one. A peptide containing only aromatic amino acids tryptophan, phenylalanine and tyrosine has a molecular weight of 11,000. The likely number of amino acid residues in the peptide. So that answer I have explained here. So molecular weight of one amino acid residue is 110 Daltons. The number of amino acid residues in the peptide, 11,000 divided by 110, that is 100. So the answer here is option 2. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. The major source of energy for the basic functioning of the cell is derived from oxidative metabolism. That is correct. Reason: mitochondria oxidize substrate to carbon dioxide, transferring high energy electron from molecular oxygen to glucose. It is not oxygen to glucose, it is glucose to oxygen. So R is incorrect. So A is true and R is false. Answer is option 3. Next one. The melting point of fatty acids is in plants derived oil is influenced by. Length and degree of unsaturation of hydrocarbon chain. Because melting temperature is directly proportional to fatty acid chain length and inversely proportional to degree of unsaturation. So the answer here is option 1. Which of the following is not a mucopolysaccharide? Inulin is not a mucopolysaccharide, it is a polymer of fructose. Then next one. Given below are two statements. In the light of the above statement, choose the correct answer from the options given below. Fatty acid synthase is an example of a tetramer of a multi-enzyme polypeptide. It is not a tetramer, it is a dimer. So first statement is not correct. Next one. Each polypeptide harbors 7 independent enzymatic functions. That is correct. So statement 1 is false and statement 2 is true. Answer is option 4. So next question. In Michaelis-Menten kinetics, what value of S as a fraction of Km is required to obtain a velocity equal to 80% Vmax? So velocity should be equal to 80% Vmax. So what you have to do here is substitute the value of value given in the options in place of the substrate concentration and find out the velocity, how much it is, whether the option which is giving you velocity equal to 80% Vmax is the correct answer. So you have the formula V0 is equal to Vmax concentration of S divided by Km plus concentration of S. So first is substitute 2 km for concentration of S, you get 0.67 Vmax. If you substitute 4 km for concentration of S, you get 0.8 Vmax, that is 80% Vmax. So the answer here is option 2, that is 4 km. If you do 0.8 km, you get 0.44 Vmax, and if you get km, then you will get 0.5 Vmax. So the answer here is option 2, 4 km. Coming to the next question, major limitation of using NMR to determine protein structure is, it is difficult to interpret the data for larger proteins, that is greater than 50 kDa. Answer is option 3. Then next one, immobilization of enzyme. Choose the correct answer from the options given below. Increases the specificity of the enzyme in batch reaction. Enzyme property does not change on immobilization. So option A statement is not correct. Lead to an increase in the apparent Km. That is correct. Makes it unsuitable for its use in a continuous reactor system. That is also not correct. Decrease the operational cost of the industrial process. That is correct. So B and D are correct. Answer is option 1. Then next one. The hormone that activates the enzyme adenylate cyclase is epinephrine. Answer is option 1. The rate of sedimentation of a particle is dependent on the. So rate of sedimentation is dependent on the size density of the particle along with the viscosity of the medium. Answer is option 4. Resolving power resolution of transmission electron microscope is of sub-nanometer level. This is because electron beams have much shorter wavelength than visible light. Answer is option 3. Which of the following represent the correct sequence of MAP kinase signal transduction pathway? RAS, RAF, ERK, MEK. Choose the correct answer from the options below. So it is first will be RAS, then RAF, then MEK, then ERK. Answer is option 2, A, B, D, C. Then next one. Arrange the following in correct sequence of step in erythrocyte cycle of malarial parasite. So first will be parasite feeds on the hemoglobin in the RBCs. Then the food vacuole of the parasite increases in size. D. Then the nucleus of the parasite comes on one side. C. The hemozoin granules are formed. E. and then parasite becomes a schizond by multiple fissions, B. So the answer here is A, D, C, E, B. Option 2. Then next one. Which of the following describes cyclin-dependent kinase? CDK is inactive or turned off in the presence of cyclin. CDK is turned on and is active in the presence of cyclin. Then CDK is present throughout the cell cycle. Yes. CDK is an enzyme that attaches phosphate groups to other proteins. CDK is a kinase, so it attaches phosphate group to other proteins. So the correct options are B and C only. Answer is option 3. Then next one. Once the cell passes the G1 checkpoint and enters S phase, the cell cycle is completed by bypassing the M phase. The cell cycle is completed by passing the G2 checkpoint. Completion of cell cycle will depend on G2 checkpoint. Answer is option 3. Completion of cell cycle will depend on G2 checkpoint. Then next one. The probability of mutation in a base pair per round of replication is 5 into 10 raised to minus 10 and the genome length of bacteria is 5 MB. The number of bacterial cells required so that there is one mutation on an average per replication cycle is. Answer is option 2, 10,000. Then next one. Fractional conversion of an immobilized enzyme continuous stirred tank reactor under steady state operation increases with an increase in agitation speed. This behavior can be attributed to. So this can be attributed to reduced external film diffusion. Answer is option 2. Then next one. In power law model of fluid rheology, if the value of the flow behavior index N is less than 1, then with increase in shear stress, the apparent viscosity decreases. Answer is option 3. Then next one. The growth pattern that you will obtain for a media containing two different carbon sources, one of which is preferentially utilized will be. So it is dioxic growth. Now dioxic means double growth is caused by the presence of two sugars in the culture media, one which is easier or preferentially utilized by the bacterium to metabolize. So during the first phase, the cells preferentially metabolize the sugar on which it can grow fast. So if it is glucose and lactose, glucose will be the first choice because it is more preferred than lactose. Only after the first sugar has been exhausted, the cells will switch on to the second sugar. That is, in case of the example I have told, it is lactose. So this is the case of dioxic growth. First it will utilize glucose. Once it is exhausted, there is a lag phase for the production of enzymes required for the utilization of lactose, and then there is an exponential phase. So the answer here is option 1, that is dioxic growth. Next one. Match the coefficients in list 1 with their corresponding downstream processing step given in list 2. Sedimentation coefficient. Centrifugation. So 1 and 2 can be eliminated, 3 and 4 we will see. Then partition coefficient, that is aqueous two-phase extraction, B1. So the answer is going to be option 4. Rejection coefficient. Rejection coefficient is ultrafiltration and activity coefficient in dialysis. So the answer here is A4, B1, C2 and D3. Option. Then next one. Match list 1 with list 2. Match the industrial application of the following enzyme. Choose the correct answer from the option given below. Penicillinase. Penicillinase is in pharmaceutical. A1. A1 is there in 2 and 3 only. 1 and 4 can be eliminated. So 2 and 3 we will see. Pectinase. Pectinase is in wine. B3. So your option is going to be option 2. Trypsin. Trypsin in leather industry and rennin in dairy industry. So the answer here is option 2. A1, B3, C2, D4. Then next one. In animal cell culture, a carbon dioxide enriched atmosphere in the incubation chamber is used. Now, why do you use carbon dioxide in the incubation chamber for animal cell culture work? That is to control the pH by generating carbonic acid. Answer is option 2. For a batch microbial process, the specific rate of substrate utilization is 0.25 per hour and the specific product formation rate is 0.20 per hour. The yield product based on the substrate consumed will be, so it will be 0.2 divided by 0.25, that is 0.8. Answer is option 1. Next one. Increasing the agitation rate in a reactor breaks large bubbles into smaller bubbles. If gas holdup does not change and the average bubble diameter reduces to half, then the gas liquid interfacial area, it will increase by 100%. Answer is option 3. Then next one, acid fast bacteria. So acid fast bacteria are gram positive, but in addition to peptidoglycan, they have an outer membrane or an envelope of the acid fast cell wall. And it contains large amounts of glycolipids, especially mycolic acids. So the answer here is acid fast bacteria have cell wall with mycolic acid. Answer is option 1. Coronavirus is positive sense single-stranded RNA virus. Then penicillin consists of a thiazolidine ring fused to a beta-lactam ring to which a variable R group is attached. This antibiotic inhibits cell wall synthesis by inhibiting glycopeptide transpeptidase. Answer is option 4. Then next one. Match items in list 1 with items in list 2. Rickettsia prowazekii. It is the causative agent of epidemic typhus. Variola major. So A2. Yeah, answer is already ready. 4. Only 2 is there in one of the options. Okay, next one. Variola major. Variola major is smallpox. Varicella zoster virus is shingles, and Borrelia recurrens is for relapsing fever. So the answer here is option 4. A2, B3, C4, D1. Then next one. Which of the following mechanism of antibiotic action is or are not correct? Tetracycline inhibits amino acyl tRNA binding to A site. That is correct. Rifampicin blocks correct position of A site amino acyl tRNA for peptidyl transfer reaction. It is incorrect because it inhibits bacterial RNA polymerase, not in protein synthesis. Chloramphenicol. Inhibition of EFTU function. That is also incorrect. It inhibits peptide bond formation. Ciprofloxacin inhibits topoisomerase. That is correct. Puromycin. Puromycin causes premature chain termination during translation, mimics 3' end of amino acyl tRNA in A site and act as acceptor. That is also correct. So which are the correct options? A, D and E. A, D and E are correct. And they have asked for not correct. So not correct is B and C. B and C only. Rifampicin and chloramphenicol. Answer is option 3. Then next one. Given below are two statements. Choose the correct answer from the options given below. The recombination frequency of genes vary from 50% to 80%. Maximum recombination frequency is 50%. It cannot go beyond 50%. So statement 1 is not correct. Statement 2. Recombination can occur anywhere along the length of the chromosome. That is also correct. So answer here is statement 1 is not correct, but statement 2 is correct. So the answer is option 4. Statement 1 is not correct, but statement 2 is correct. Then next one. Match items in list 1 with items in list 2. Silent mutation. Silent mutation amino acid sequence is unchanged. A4. A4 is there in 2 and 3. 1 and 4 can be eliminated. Missense mutation. Missense mutation amino acid sequence is changed in one amino acid. B3. So your answer is going to be option 3. Next one, nonsense mutation. Amino acid codon is changed to a stop codon. That's C1. Frame shift mutation. Amino acid sequence beyond the site of mutation is changed. So the answer here is option 3, A4, B3, C1 and D2. Then next one, given below are two statements. Sickle cell trait protects humans from malaria. That's correct. Sickle cell trait can be transmitted even if both the parents are heterozygous for the beta globin gene. That is also correct. So statement 1 and 2 are correct. Answer is option 1. Then, in which of the following genotypes of lac operon the enzyme synthesis would be constitutive? So if you see B and D, here operator is constitutive in both the partial diploids. Operator is constitutive. So repressor cannot bind and the enzyme synthesis become constitutive. Then next one is D. In case of D, the repressor itself is defective. So repressor cannot bind to the operator. So the synthesis of beta galactosidase enzymes will become constitutive. So the answer here is option B and D. Option 3 is the answer. Next one. Assume that height in a plant is controlled by 3 genes A, B and C, and each additive allele contributes equally. The base height of the plant is 20 cm and the maximum possible height is 38 cm. The height of a plant with the genotype A, small b, capital C, small c will be. So A, B, C, 3 genes and all have equal contribution. Maximum possible height is 38 centimeter. So capital A, capital B, capital C will be 38 centimeter and the contribution of each gene will be 38 divided by 6, that is 6.33. So A contributes 6.33 and small a contributes half of what A contributes. So that will be 3.165. So plant height with genotype A, small b, capital C, small c is. So wherever capital A or any gene with capital letter comes, you have to put 6.33 and wherever small letter comes, you have to put 3.165 and then add everything, you will get answer as 29. So answer is approximately 29 centimeters. Answer is option 2. Then next one, based on this phylogenetic tree, which statement is not correct? The salamander lineage is a basal taxon. That is correct. No branching before salamander is there. Salamanders are sister group to the group containing lizards, goats and human. That is also correct. Salamanders are closely related to goats as to humans. That is also correct. Lizards are more closely related to salamanders than humans. Lizards are more closely related to goat and human and not to salamanders. So they have asked for the not correct statement and option 4 is not correct. So the answer is option 4. Then next one. In the human ABO blood group system, the alleles A and B are dominant to O. What will be the number of different possible genotypes? So you will get 6. How? There you will get capital A, then A, O, B, 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 O, O, and A, B. So there will be 6 possibilities. Then next one. The stop codon lead to termination of translation because there are no tRNAs corresponding to stop codon. Answer is option 1. Then next one. In a family, if the father is affected by X-linked dominant trait and mother is normal, then So this is the father, he is having X-linked dominant trait, and this is the mother. So you will get a daughter who is deceased and none of the sons will be affected. So answer here is option 3. All daughters and none of the sons will be affected. Answer is option 3. Then next one. Which one of the following is a protein prediction server based on artificial intelligence? It is AlphaFold. Answer is option 2. Then given below are two statements. In the light of the above statement, choose the appropriate answer from the options given below. Statement 1. BLAST compares protein as well as nucleotide sequence in order to identify areas of similarity. That is correct. GenBank is a repository of data on the phenotypic results of gene knockouts in humans. It gives genotypic information. So it is not phenotypic results, it is genotypic results. So the answer here is option 3. Statement 1 is correct, but statement 2 is incorrect. And next one. RNA viruses are entirely dependent on the host for protein translation machinery. Answer is option 4. RNA virus replicates in cytoplasm. Answer is option 1. The following statement is not true for the receptor binding site of a virus. Binds specifically to a cellular receptor, cannot bind to neutralizing antibodies. Answer is option 2. The statement is not true for receptor binding site of a virus. Then next one. Which of the following hypersensitivity reactions involve the complement activation? So it is both type 2 and type 3 hypersensitivity reactions. Answer is option 4. Which of the following phagocytic cell is not a tissue-specific macrophage cell? It is dendritic cell. Answer is option 4. The next one. Which of the following statement is not true about myeloma cells used in the hybridoma technique for the production of monoclonal antibodies? Answer is option 4. They can produce antibodies. Then which of the following is an inactivated vaccine? So it is Covaxin. Answer is option 3.